So as Lily mentioned, my lab recently moving from the University of Michigan to Zhejiang University at Hangzhou is interested in ion channels in the lysosome. So the primary function of lysosome is degradation, which takes place in the lysosome lumen. But the degradation is regulated by cellular signals, like when the degradation should take place, when to finish, how hard the lysosomes should work, such and such. So as membrane proteins, so ion channels in the lysosome, so as membrane protein in the lysosome, can bridge or communicate between the lysosome lumen where the degradation takes place and the cytosol where all the signaling takes place. So to study how lysosome degradation is regulated, some years ago, actually 15 years ago, we developed the lysosome patch clamp technique. So in this method, we enlarge the lysosomes with small molecules like a vacuolin. And so now we have other ways to enlarge lysosome as well. Then we cut the cells open and let the lysosomes release into the dish. Now we can perform patch clamp experiments directly on these isolated lysosome. So identity of the lysosome can be conferred by LAMP1, which is lysosome associated membrane protein 1 that's only present in the later endosome and lysosome. So using the lysosome patch clamp technique, we have found a bunch of conductances in the lysosome and molecularly identified a couple of them. So for example, trypamyl mucolipin trip channels, calcium channels in the lysosome, 2'-TPC proteins, sodium selective channels in the lysosome, BK channels, potassium channels in the lysosome, lysovarex channels, chloride channels in the lysosome, and I will be talking about proton channels in the lysosome today. So we know lysosome lumen is highly acidic, so there's almost 1,000-fold proton concentration gradient across the lysosomal membrane, which is established by these VATPase, right? So when you inhibit a VATPase, like using, say, bufferomycin A1, lysosomes are very quickly deacidified. So therefore, there must exist proton leak conductors in the lysosome. So let's just call it a lyso-H. So lyso-H together with the VATPase will set up this pH optimum, roughly pH 4.5 to 5.0. So this optimal is preferred by all those enzymes, most enzymes in the lysosome, because these enzymes work much better at this pH. So, so when the lysosome pH is too high, the VATPase will pump the proton into the lysosome lumen, and when the lysosome's lumen is over-acidified, lyso-H will presumably leak out the protons, right? So this is the classic negative feedback loop. Yeah. So, so we want to study we want to calculate lyso-H, and so in the whole lysosome patch clamp recording, when we created a gradient, acidic in the lumen and neutral at the cytosol site, we and the physiological lysosomal pH, let's say pH 4.6, we can see some currents in some lysosomes. But when we lower the pH to 3.5, and all lysosomes have large currents. So, So the proton gradient is the only thing that matters. So other cations like sodium, potassium, such do not affect the current. So this is presumably lyso-H current. And all cell types we look at, HEK cells, human fibroblast, primary neurons, all cell types have this lyso-H current. So we want to find the protein responsible for lyso-H. So we quickly rule out some candidates in the literature like CLC and 7, tryptophan 1 channel, TPC 1 channel, and then we did the screening with 2 lysosome membrane protein libraries, and one of the libraries, knockout library for loss of function studies, and the other one is for the fusion construct for the overexpression study. So anyway, to make a long story short, we have one hit, which is PMN175, or we call it LAPAP, and PMN175 fusion proteins are localized mainly in the lysosome, a little bit in the early endosome, but not in the mitochondria. So when we enlarge the lysosome with vacuolia, you can see that PMN175 protein is localized in the perimeter membranes of lysosome. Because we don't have good antibody for endogenous PMN175 protein, so we generate the HAA knock-in cells. You can see that endogenous PMN175 proteins are co-localized with LAMP1. So PMN175 is required for the lyso-H current. You can see knockout is very clean. So PMN175 is probably the primary, if not the solo lyso-H current in the lysosome. On the other hand, overexpression of PMN175 can dramatically increase the lyso-H currents. And more importantly, so endogenous PMN175 protein probably is strictly lysosomal protein, because in the whole cell recording, we do not see any proton current. But when we overexpress PMN175, we see large currents. We see large proton currents. So therefore, PMN175 is both necessary and sufficient for lysosomal proton currents. So how do we know this current is proton current and not a proton activated current? So when we replace all the physiological anions and cations like chloride, sodium, or potassium with large non-permanent ions like e-IDG or gluconate, this current is not affected. On the other hand, when we change the pH, the reversal potentials change dramatically. So this is likely a proton current. So when we performed this pH reversal potential analysis, this plot fits very nicely with a slope of 53 millivolts per pH unit. So based on GHK or Nernst equation, this must be highly proton selective channel. But when we put the potassium in the computation, and so this is the standard protocol to study ion selectivity of channels, but the slope is still roughly the same. So when we calculate the selectivity, both based on this slope analysis or the reversal potential based on measurement based on under so-called bionic conditions, we find that the channel is roughly 50 to 100,000 times selected for proton over sodium or potassium. So although there is a significant difference between sodium and potassium, I will explain that a little bit later. But these values are comparable to those by known plus membrane channel, the proton channel, like the OTOP1, which is the sour taste channel in the tongue. So, so even though the channel is selective, right? So because of sodium and potassium concentrations are much higher. They are much more abundant in the cells in the lysosome. So if we calculate the so-called fractional current by taking into consideration of these ion concentrations, proton current is still dominant in the native lysosome. So, so we also use the pH imaging to confirm that the channel is indeed a proton permeable. So here we use the proline as the pH indicator. We can, so the movie is already in play. Let me play again. So this is P175 transfected cells. You can see the proline is quenched very rapidly. So here we use the ammonium chloride, acetate or ammonium chloride as a positive and negative controls respectively for this experiment. So this is the statistics here. So if P175 is indeed the proton leak, we would expect the lysosomes are over-acidified or hyper-acidified in the knockout cells. That seemed to be the case. And when we use the lysochecker to stain lysosome, you can see the lysochecker staining is higher in the knockout cells. But of course, another possibility is they are just more lysosomes in the knockout cells, as in many lysosome storage disease cells. But however, LAMP1 staining is comparable for wild-type knockout cells. So therefore, it's likely that the proton concentration is high in the knockout cells. But lysochecker can only provide a very rough estimate for lysosomal pH. So we therefore use the 3 sets of ratiometric dyes to measure the lysosomal pH. So Joe Swanson is our collaborator who's expert in lysosome pH measurement, did some blind experiments here. And when we block the VATPs, use different concentration of barfomycin, lysosomes are deacidified, but there are still differences between wild-type and knockout cells. We find that a very low concentration of barfomycin, 1 nanomole, can bring the lysosomal pH to normal in the knockout cells. So, so this is so-called lysosomal pH normalization or correction. So I will talk about the significance of these in a few slides. So looks like P175 is a proton leak channel in the lysosome. But is this indeed the leak channel, meaning the channel is constitutively open all the time? So this is actually not an easy question to answer, because when you see more currents, when you drop the pH, it could be more permeable proton ions, or could be more driving force, or more channels activate. So if we assume the channel is a leak channel, we could do one experiment to test this. We can simply just reverse the proton gradient. That means you pH 4.6 in the cytosolic side, but the pH 7.2 in the luminous side, and in the lysosome recording. So this is so-called reverse the pH experiment. So by the way, if the channel, if the open all the time, so in this reverse pH gradient experiment, one would still expect the current, but the current should be the outward current now, not the inward current. Does that make sense? So, but we don't see that, right? We don't see any current at in this reverse pH gradient experiment, even though there's a huge proton concentration gradient. But when we change the luminoside pH to 4.6, we start to see large current. But this current reverses at 0 millivolts because now we have symmetric proton gradient. So we conclude the channel is activated or gated by luminoside protons. This is why we initially call Lysosome as a proton activated, proton permeable channel, light pipe. We next wonder whether there are some chemical agonists for the channel. So we search the literature, find out that arachidonic acid is reported to cause proton flux in isolated lysosome in some early studies. So we found that arachidonic acid actually can indeed the active channel. But remember, in the native lysosome, pH 4.6 may or may not activate the channel, but arachidonic acid can always reliably activate the channel. We also found some synthetic compounds can activate channel, DCPIB or ML6733. These compounds known to modulate other channels. We also now did the high throughput screening to find lots of highly potent synthetic agonists for the channel. So when we use these synthetic agonists, we can now clearly see that in this reverse proton gradient experiment, the channel is gated. The current is purely outward. So, in the lysosome, we can now measure the agonist-induced proton release from the lysosome lumen. And so here we use lysosome target genetic encode pH indicator by fluorine directly T175. And you can see that agonist application, we can see the proton release from lysosome with a bunch of control experiments to show that this is the proton releases from lysosome. For example, we use GPN to abolish the proton gradient, then that effect is gone. So this is the overexpressed T175 protein. In the endogenous setting, we can also see proton release, but it's much smaller. But again, we did a control experiment to show that this is the release from the lysosome. So using the lumino dyes, now we can see arachidonic acid can induce the reduction of luminoprotons in Y-type cells, but not in the knockout cells. So, well, I can stop my presentation here, but there's a problem here. That's because T175 was reported to be constitutively active potassium current by Dr. Degenren at UPenn. So while we both agree this is a lysosomal channel, we have at least two controversies. One is whether the channel is constitutively active or gated. The other one is the selective is potassium or proton. So it's important to point it out in Dr. Ren's studies, as well as several follow-up studies that they use the neutral pH for their recordings. And we know neutral pH is physiological for plasma membrane channels, but not for the lysosomal channels. So when we use the potassium in our recorded solution at the luminoside pH 7.2, we can see a little bit of basal currents in the T175 overexpressing cells. But we can then activate the current with arachidonic acid. And when we switch to low pH solutions, the reversal potential changes dramatically. So we think the channel is not constitutively active, but for both for proton and potassium current. So, and this point is very clear in the endogenous setting in the overexpressed cells. So, so we know that in the overexpression system, sometimes you might see some basal current, but in the native lysosomes, we almost never see basal potassium currents, but we can activate the current with arachidonic acid or synthetic agonist. So this is also very clear. We don't see much basal potassium currents, but with low pH, we can see large proton current. So 175 can be potassium or cesium current, but it's gated, right? Because we cannot see the potassium current in the lysosome, and but you can see proton current as far as the pH is low. We think in native lysosomes and the physiological pH, T175 functions as a proton channel. So what's going on? So when we pick this, we pick the cells with large basal current in some overexpressing T175 overexpressed cells, we can see proton indeed can inhibit the potassium current. So at this point, a paper came out from Science Advance by Kim Min-Fu's lab at Ohio State University. This is several weeks before our paper was officially accepted. So their results are very similar to our overexpression studies. So T175 current is inhibited by low pH and it's proton permeable. So we will partially scoop it, but in their study, they also saw the structures at both neutral pH and the low pH, and find out in their structure at the low pH, the channel pore is narrowed, so that the dehydrated potassium cannot pass anymore, right? So here's an analogy. So for voltage-gated calcium channels, which are very selective for calcium, sodium or potassium really cannot pass under physiological conditions. But when you remove all the calcium, that the sodium and the potassium can actually pass happily, probably even more happily than calcium. So T175 can be a potassium channel, but in the lysosome, proton beats potassium. So at this point, we find out that one amino acid, D41, might be responsible for proton permeation or selectivity or activation, because this mutant, the proton current is abolished, but the potassium current, which requires synthetic agonist for activation, is intact here. So we find, so unlike wild type channel, this D41 mutant cannot rescue the lysosomal pH phenotype seen in the knockout cells. And so optimal pH is required for the activity of the luminal enzymes like cathepsin B, cathepsin D. So again, in this experiment, the loss of activity, cathepsin B activity, shown by magic red staining, can be rescued by Y type construct, but not by D41A mutant. So same is true for the cathepsin D activity when we perform the lyso pH correction experiment I mentioned earlier, using just 1 nanomole of bufferomycin. So the activity is rescued. So in summary, we believe that T175 is important for lysosome function, and its proton conductance is primarily responsible. So finally, the T175 mutation is a high risk for Parkinson. And so we generated knockout mice and found that in the mouse neurons, Human 175 behave just like our studies using human cells in terms of a steady state pH and lyso-H current and agonist-induced pH change, lysosomal proton release, and the cathepsin B and the cathepsin D activities. So cathepsin B or D enzymes are known to regulate alpha-synuclein protein aggregation degradation. And so in the mouse brain, we find that alpha-synuclein aggregate accumulation is much more obvious in the knockout cells if we give sun challenge, even in young mice. And they also behavioral phenotypes as well. So I will stop my presentation here. So this is the work that was done by mainly by two postdocs in the lab, Mei Chen and Ping, and a graduate student, Ce in the lab. So I will stop here. Thank you very much for listening.